Hi everyone, this is part 1 of the Mod 15 lecture videos on regulation of gene expression. Mod 15 covers chapter 11 in the textbook, which is titled How Genes Are Controlled. An introduction to chapter 11. The first mammal to be cloned was a sheep in 1996. Her name was Dolly, and it was a really big deal at the time, the first successful cloning of a mammal. Nowadays, you can even clone your pet if you wanted, although it comes with a hefty price tag. These two beagle puppies here were cloned from an adult beagle, but you can expect it to cost anywhere from $50,000 to $75,000 if you would like to clone one of your pets. So the cloning of Dolly the sheep in 1996 was a huge event. However, the outcomes of animal cloning have not quite lived up to initial expectations. And that's because cloned animals are often abnormal with shortened lifespans and increased incidences of age-related diseases that happen at younger ages. So Dolly the sheep that was cloned in 1996, the typical lifespan for her breed of sheep is 12 years old. However, Dolly passed away at the age of 6 from a lung disease that is typically not seen until the sheep is much older. So it was an age-related disease that affected a 6-year-old sheep that should not have affected a sheep that young. So the cloned animals tend to have some health issues in that respect. The difficulties in producing a healthy clone highlight the complex role genes play in the growth of an organism. And we're going to talk about how genes are controlled in this module. So we've already talked about the genetic basis of cancer, so we will not be covering that. In this module, we'll be focusing on the actual control of gene expression, and then at the end, we'll talk a little bit about the cloning of plants and animals. So control of gene expression. We're going to talk about control of gene expression in prokaryotes first, because prokaryotes, it's a bit simpler how it works in prokaryotes versus eukaryotes. It becomes a bit more complex. The overall process by which genetic information flows from genes to proteins, that is from genotype to phenotype, is called gene expression. This is a little bit of a review from Mod 14. Gene regulation is a new vocab word, and that means the turning on and off of genes. Gene regulation can help organisms respond to environmental changes, especially prokaryotic organisms like bacteria, because a lot of times they're exposed to a wide variety of environmental conditions. Think about the E. coli living in your large intestine or small intestine. At any point in the day, depending on what you eat, the nutrients that they are surrounded by are going to change directly based on what you are eating. So they have to be able to respond to any of those different possible environments based on what you ate. So proteins interacting with DNA turn prokaryotic genes on or off in response to environmental changes. This is how prokaryotes are able to deal with environmental conditions that can vary so much. In prokaryotes, genes for related enzymes are often controlled together in units called operons. We are going to talk about a specific prokaryotic operon called the lac operon. In this image here, you'll see the operon shown by this bracket. It includes the promoter, the operator, and these 3 different lactose utilization genes. And then regulatory proteins, such as this repressor here, bind to control sequences in the DNA to control whether or not an operon is expressed. On the previous slide, I mentioned that E. coli lives in your small and large intestine. It's exposed to constantly fluctuating environmental conditions depending on what you just ate. The lac operon here has lactose utilization genes. So lactose, remember, is the sugar that is found in milk. These 3 genes allow E. coli to digest lactose when it is present in the environment. However, if lactose is not present in the environment, so if you have not consumed any milk recently, it's detrimental for E. coli to have these lactose utilization genes turned on because it would be a waste of energy to produce enzymes that can digest lactose when there's no lactose in the environment. So each one of these lactose utilization genes codes for a specific enzyme that will help E. coli digest lactose. On the next slide, we're going to look more specifically at the lac operon and see what it looks like when it's turned off and when the operon is turned on. Also, just a heads up, your homework question for Mod 15 is going to heavily focus on the lac operon and the discussion board. So at the top, we have the operon turned off, and at the bottom, we have the operon turned on. The operon is turned off when lactose is absent from the environment because, again, it's energetically detrimental to E. coli to be producing lactose-utilizing enzymes when there is no lactose present in the environment. So we have our lac operon here, which consists of the promoter, the operator, and the 3 lactose utilization genes. The promoter in green is where RNA polymerase needs to bind to transcribe this DNA into mRNA. This regulatory gene in blue is not part of the operon. The regulatory gene codes for the mRNA that then codes for a specific protein. This protein is an active repressor protein, and when lactose is absent from the environment, this active repressor will bind to the operator in yellow of the lac operon. When the active repressor is bound to the operator, notice how it blocks part of the promoter as well. Because of this, RNA polymerase cannot attach to the promoter, so these lactose utilization genes cannot be transcribed into mRNA and then translated into the protein product, the enzymes that help digest lactose. So the lac operon is turned off when lactose is absent. Here in the bottom, this is what happens when lactose is present in the environment. So up here, this is what happens when you have not consumed any milk for a long time. Down here, let's say you just drink a big glass of milk and now the E. coli in your small intestine are being exposed to lactose. So when lactose is present, we still have the regulatory gene in the DNA that codes for the mRNA and then is translated into the repressor protein. However, the repressor protein, when lactose is present in the environment, lactose is shown by this little pink circle. When lactose is present, it will bind to the receptor or the repressor and change its shape. Once the shape of the repressor is changed after it is bound to lactose, it is no longer active. It has been inactivated, and so that repressor will no longer bind to the operator. The shape has changed and it can no longer bind to the operator. When the repressor is not at the operator, the promoter is no longer blocked, so now RNA polymerase can bind to the promoter and initiate transcription to make this mRNA sequence. This mRNA sequence can then be translated into the 3 specific enzymes from the 3 lactose utilization genes that are needed to help E. coli digest lactose. So when lactose is absent, the operon is turned off because the repressor binds to the operator. When lactose is present in the environment, it inactivates the repressor and the operon is turned on. So the operon is producing the lactose utilization enzymes only when lactose is present in the environment. The lac operon, this type of operon is known as an inducible operon because its normal state is to be turned off, but it can be induced or turned on when lactose is present in the environment. The reverse type of operon, so the reverse of an inducible operon, is a repressible operon. A repressible operon is normally on but can be turned off in certain situations. The TRP or the trip operon is an example of a repressible operon. TRP stands for tryptophan, which is an amino acid. The trip operon is also found in E. coli, and the normal state of the trip operon is to be turned on. The trip operon produces mRNA that then will produce the amino acid tryptophan, so its normal state is to be turned on. However, if there is tryptophan in the environment, E. coli prefers to use the tryptophan from the environment versus making its own, so it will then turn off the trip operon if tryptophan is present in the environment, because it's more energy efficient for E. coli to get tryptophan from the environment than from making it itself. So the normal state of the trip operon is turned on. So when tryptophan is absent, when there's no tryptophan, the repressor is inactive, and so the operon is turned on. E. coli can make its own tryptophan. When tryptophan is present in the environment, E. coli doesn't want to make its own tryptophan. It's not energy efficient to do so. It can just take it from the environment. So tryptophan actually activates the repressor and turns off the TRP operon. So when tryptophan is present, the repressor is activated and turns off the operon. So the TRP operon is an example of a repressible operon. It's normally on but can be turned off. And then the LAC operon over here, that's an example of an inducible operon. It's normally off but can be turned on. So just a quick refresh of the LAC operon. When lactose is not present in the environment, the operon is turned off. The active repressor binds to the operator, which blocks RNA polymerase from binding to the promoter. However, when there is lactose in the environment, lactose will bind to the repressor and inactivate it so that it can no longer bind to the operator. And then RNA polymerase will be able to bind to the promoter and transcribe those sequences that code for the lactose utilization genes. So a checkpoint question based on the LAC and the TRP operons. A certain mutation in E. coli impairs the ability of the LAC repressor, which is the repressor protein, to bind to the LAC operator. How would this affect the cell? Take a second and think about this, and then we'll come back together. So hopefully you were thinking something along the lines of the cell would produce the enzymes for lactose metabolism continuously, even when lactose is not present, thereby wasting energy. So it's in the cell's best interest to not produce those enzymes when lactose is not present. But if that mutation was there, the cell would no longer be able to regulate that. Now we're going to start talking about examples in eukaryotic cells. So the LAC operon and the TRP operon, they are in prokaryotic cells. Prokaryotic cells have a little bit simpler mechanisms of the start of transcription, the operons like we just talked about. Now we're going to start talking about eukaryotic cells. Chromosome structure and chemical modifications can affect gene expression. As a zygote develops into a multicellular organism, so a zygote is when an egg and sperm come together and fertilize. That then will develop into a zygote, and then a zygote develops into an embryo, which develops into a multicellular organism. As a zygote develops into a multicellular organism, individual cells must undergo differentiation, which means they become specialized in structure and function. You can see a few examples of differentiated cells in the human body here. We have stem cells, bone cells, blood cells, muscle cells, endothelial cells, nerve cells, skin cells, and fat cells. Those are just a few examples. Each cell type must maintain a specific regimen of gene expression in which some genes are expressed and others are not. So the difference between these different types of cells are not due to different genes being present, but instead they're due to selective gene expression. To put that in a simpler term, all of these different types of cells in your body have the exact same DNA, all the exact same. However, different genes are expressed in your stem cells versus in your bone cells, and different genes are turned on in your blood cells versus in your muscle cells. Hopefully that is making sense. So the differences between cell types are not due to different genes being present. They all have the exact same DNA, but instead due to only selected genes being expressed to make a certain cell a muscle cell or to make a certain cell a skin cell. A chromosome contains DNA wound around clusters of small proteins called histones, which forms a string of bead-like nucleosomes. The reason why we're talking about this after that previous slide is because this is one of the ways that eukaryotes can control what genes are expressed and what genes aren't. Prokaryotes don't tend to have as much DNA, so this isn't as efficient of a way for them. So in eukaryotic DNA, we have our DNA double helix, which has about a 2 nanometer diameter. So it's teeny, teeny, tiny. The typical DNA in the eukaryotic cell is thousands of times longer than the diameter of the nucleus, so it has to be really tightly packed to fit into the nucleus. This is how that DNA is tightly packed. The more tightly packed a gene is, the less likely it is that it will be expressed. So say if we look at a duplicated metaphase chromosome over here, if there's a gene in the middle of all these other genes, it's very unlikely that it will be expressed because the transcription factors in RNA polymerase just will not be able to get to the gene to create an mRNA transcript. But we're going to look at really quickly how the DNA packs that tightly. So we start out with our 2 nanometer diameter DNA double helix. That DNA double helix then wraps around these histone proteins. In fact, it wraps around a series of 8 histone proteins, 1, 2, 3, 4, 5, 6, 7, 8, to become a beads on a string model. So the beads are these complexes of histone proteins with DNA wrapped around it, and then they're joined to other beads through these linker strands of DNA. If you look at the beads on a string representation under a microscope, you can see the beads in those circular areas and then the little thin DNA linkers in between them. The beads on a string are also called nucleosomes. So those 8 histone proteins and the DNA wrapped around them is called a nucleosome. A nucleosome is 10 nanometers in diameter. Those nucleosomes are then tightly wound together to create this tight helical fiber, which is made up of that beads on a string model tightly placed together. That has about a 30 nanometer diameter. And then this tight helical fiber is combined into this looped domain. So it's packed even tighter. The looped domain has a 300 nanometer fiber. So these are getting bigger and bigger as we go along. And then the actual duplicated chromosomes are made up of those looped domains. The diameter of a single chromosome is about 700 nanometers. So by the time the genes are this tightly packed, if a gene is way on the inside of this chromosome with piles of looped domains on top of it and on every which side of it, it's very unlikely that that gene will be able to be expressed, again, because RNA polymerase will not even be able to get to the gene. So how tightly packed the DNA is can influence what genes are expressed and what aren't. DNA packing tends to block gene expression by preventing access of transcription proteins to DNA, which is what I was just talking about with RNA polymerase. How tightly packed the DNA is can be modified. Chemical modification of DNA bases or histone proteins can result in epigenetic inheritance. So there's something called DNA methylation, which is when methyl groups get added onto the DNA. A certain amount of DNA methylation is needed for normal embryonic development, but the addition of the methyl groups does tend to open up the DNA a little bit. So if you get too much DNA methylation, certain genes might be exposed that were previously not exposed. So it can result in new genes being expressed. And this methylation of the DNA is passed on from a cell to all offspring of the cell, and also passed on from parent to offspring. So this is known as epigenetic inheritance. Epigenetic inheritance is traits that are inherited that are not directly related to the nucleotide sequence of DNA. So the epigenetic inheritance is due to the methylation of the DNA, not to the actual nucleotide sequence. It's a really cool, newly developing area of research. We still don't know too much about it, but if you are interested, I would encourage you to do some Googling on it because it's a fascinating topic. Another example of DNA packing that I want to talk about is X chromosome inactivation. X chromosome inactivation occurs in the cells of female mammals. A very common example of X chromosome inactivation is the tortoiseshell cat. That coat color is a direct result from X chromosome inactivation. We are going to talk about this more now. So we know that a male has chromosomes X and Y, whereas a female has two X chromosomes. Because a female has two X chromosomes, one of them needs to be inactivated, or else females would produce double the amount of proteins or enzymes for any genes on the X chromosome than a male would, which is not what happens. So in females, one of the X chromosomes in every single cell is inactivated, and this is random. So it doesn't, in some cells, the X chromosome from the mother could be inactivated. In other cells, the X chromosome from that organism's father could be inactivated. It's completely random, but one X chromosome per cell will be inactivated because you don't need protein products from two X chromosomes. You only need one. The inactivated X chromosome balls up and becomes super small. You can see the two inactive chromosomes there. We call them a Barr body, B-A-R-R. The inactive X chromosome is known as a Barr body, and since it is so tightly packed, none of those genes are expressed. We're going to talk through this example here. So we have the early embryo of this tortoiseshell cat on the right. It has two X chromosomes. It's a female. This allele for orange fur, let's say that came from its father, and then this X chromosome has the allele for black fur that came from its mother. So it has an allele for orange and an allele for black fur. However, since it's a female, it has two X chromosomes. One of those X chromosomes in every cell of the body will be inactivated, but this will be random. So certain cells of the body might have the X chromosome with the allele for orange fur inactivated, and then other cells of the body might have the X chromosome with the allele for black fur inactivated. It is very random. So as a result of cell division and that random X chromosome inactivation, in this adult tortoiseshell cat, there are two different possibilities for cell populations. You could have cells where the active X chromosome is the X chromosome with the orange fur allele, and the inactive chromosome is the black fur allele X chromosome. Or you could have the reverse, where the active X chromosome is the chromosome with the allele for black fur, and the inactive X chromosome is the chromosome with the allele for orange fur. And that is what causes this coat color pattern in the cat. So all of the cells with the active allele for black fur will be black, and then all of the cells with the active X with the orange fur allele will be orange. This is why you typically only see this coat pattern in females, because in order for a cat to be this color, it needs two X chromosomes. You could theoretically see this coat color in a male. However, it would mean that he had two X chromosomes and a Y chromosome, so he likely would be sterile because his genotype would be XXY. You need the two X chromosomes to see the X chromosome inactivation that causes this tortoiseshell fur pattern. So a checkpoint question: in your body, a nerve cell has a very different structure and performs very different functions than a skin cell. Because the two cell types have the same genes, how can these cells be so different? Hopefully you thought back to cell differentiation and how we talked about how all cells in your body have the exact same genes, but certain genes are turned on depending on the type of cell. Each cell type must be expressing certain genes that are present in, but not expressed in, the other cell type. That's how that would occur. So we talked about how transcription is controlled in prokaryotes, specifically with the lac operon and the trip operon. Now we're going to talk about how transcription is controlled in eukaryotes. It's a bit more complex in eukaryotes. And I will say this is probably one of the most important regulation of gene expression points, is the control of the start of transcription. Complex assemblies of proteins control eukaryotic transcription. A variety of regulatory proteins interact with DNA and with each other to turn the transcription of eukaryotic genes on or off. This image down here shows the variety of regulatory proteins. We have RNA polymerase, which we know is required for transcription to occur. We have a DNA bending protein. We have these green activator proteins, and then we have these other purple proteins. These activator proteins and then this group of other proteins are called transcription factors. Transcription factors are proteins that promote the binding of RNA polymerase to a specific gene. Your discussion board is going to ask you to compare what we're about to talk about on the next slide with the lac operon regulation. So just be aware of that. It's going to ask you to draw a eukaryotic gene in the presence of transcription factors, which is what this image shows. So again, we have all those proteins we just talked about, the transcription factors, which are the green activator proteins, and the other purple proteins, the DNA bending protein, and then RNA polymerase. In eukaryotic DNA, you typically have a gene shown over here in blue, a promoter right before the gene, and then far away but on the same chromosome, you'll have enhancers, which are where the activator proteins actually bind. These enhancers are shown in yellow. In order for eukaryotic transcription to start, these green activator proteins have to bind to the enhancers that are located upstream of the actual gene that needs to be transcribed. These activator proteins will then recruit other transcription factors that will then help attach RNA polymerase to the actual promoter. And then a DNA bending protein is needed right here to actually bend the DNA so that the two enhancers can be directly above the promoter and then the gene of interest. So the two activator proteins will bind to the enhancers, the DNA bending protein bends the DNA, and then the other transcription factors help facilitate the attachment of RNA polymerase to the promoter, and all of these are attached to those two activator proteins that are bound to the enhancers. What must occur before RNA polymerase can bind to a promoter and transcribe a specific eukaryotic gene? It's everything we just talked about. The transcription factors must bind to enhancers to facilitate the attachment of RNA polymerase to the promoter. A lot of times transcription factors are very specific to their specific gene. And so if any of the transcription factors aren't present or the DNA bending protein isn't present, transcription cannot occur. We're now going to talk about another way that organisms can regulate their gene expression. So eukaryotic RNA may be spliced in more than one way. This is RNA processing. We talked a little bit about RNA processing in the last module. This video is a quick reminder about what goes on during RNA processing.
As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus.
That was a refresher on the last module. Remember, a cap and a tail is added to the RNA, and then any introns are spliced out so that only the exons are left in the mRNA transcript. Alternative RNA splicing means that the exons can be spliced together in more than one way. After transcription, alternative RNA splicing may generate two or more types of mRNA from the same transcript. In humans, more than 90% of protein coding genes appear to undergo alternate splicing. So this really increases the sheer number of different enzymes or gene products that we can make. This here is an example of alternative splicing. So we have the DNA shown up here in blue. Remember, the introns are this lighter blue. Those are the non-coding regions of the DNA. And then we have the exons in darker colors of blue. We have exons 1, 2, 3, 4, and 5. Those are the coding regions of DNA. The mRNA that is a result from this DNA, we have the RNA transcript. This one still has the introns within it, and it has those exons 1, 2, 3, 4, and 5, and then it has that cap in the tail. This RNA transcript can be spliced a variety of different ways. One way will result in the mRNA transcript having the 3 exon. The other way will result in the mRNA transcript having the 4 exon, but not the 3. So in this left one, the mRNA, the introns were spliced out, and then exon 3 was spliced in, but exon 4 was not. And then in this right mRNA transcript, exon 3 was spliced out along with the introns, and then only exon 4 remains. So this is alternative splicing. This is from the same gene in DNA, but there are different ways that that mRNA can be spliced together when it's being modified. This is just a bigger image of that same figure we just talked about. How is it possible that just under 21,000 human genes can produce more than 100,000 polypeptides? Well, it's possible because of alternative RNA splicing. So through alternate splicing, each kind of polypeptide is encoded by an mRNA molecule containing a different combination of exons. So in this image, this one gene here produces likely two different proteins. So 2 different mRNA transcripts, which will be translated differently into different protein products. So alternative splicing is how our genome that only has 21,000 genes can still code for more than 100,000 protein polypeptides. Later stages of gene expression are also subject to regulation. One of those is the lifetime of the messenger RNA molecule. The lifetime of a messenger RNA molecule helps determine how much protein is made, as do factors involved in translation. Some mRNA molecules are degraded really quickly within minutes after they are made. Others, such as the mRNA that specifies hemoglobin, that stays around for almost a month in our cells. So different factors about the mRNA dictate how long it stays in the cell. Obviously, if an mRNA stays in the cell longer, a lot more protein product is going to be made than if it only stays in the cell for a minute or 2.
The lifespan of mRNA molecules helps determine how much of a protein is made. Nucleotide sequences that affect mRNA longevity are often found in the 3' untranslated region following the stop codon. Enzymes attack the poly-A tail first, and eventually the rest of the mRNA. Depending on its ability to resist this degradation, a eukaryotic mRNA molecule may last from hours to weeks. The longer an mRNA lasts, the more protein is made.
So longer-lived mRNA has the ability to resist degradation, and the longer it stays around, the more protein product is going to be made because translation can occur on that mRNA sequence more than an mRNA sequence that was very short-lived and degraded quickly. Some other later stages of gene expression that can be regulated are the activation of a protein. So a protein may need to be activated in some way before it's functional, and then eventually the cell will also break it down. So activating the protein is one way, and how long the protein is activated until it's degraded is another way that gene expression can be regulated. So this is an example of insulin. The initial polypeptide that forms insulin is this long strand. However, in order for it to become the active insulin, it needs to be cleaved or cut at these 2 cleavage sites. After it's cut at those 2 cleavage sites, the polypeptide will then fold into its correct orientation and form these SS linkages. This 2-part polypeptide is the active form of insulin that can actually do the correct function within the human body. Proteins also, some other ways that they need to be activated. They might need to be phosphorylated. They might need to be transported to a specific location. So all of those are things that regulate when a protein becomes active. And this video does a nice job of summarizing some of those different ways.
Often, polypeptides must be modified in some way to produce functional proteins. For example, a polypeptide is cleaved in 2 or more polypeptide chains. Other molecules might be attached to the protein, or the protein might be phosphorylated. Some proteins need to be transported to specific destinations.
So this last example was a protein being transported. This certain protein may not ever become active until it is transported to its target destination organelle. So if the cell doesn't want that protein to be active, it would just never transport it to its target location. Protein degradation also influences gene expression in a cell, because if a protein is around for a long time, it will have a lot more impact in the cell versus if it's only around for a short time, it will have less of an impact.
A cell can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tagged protein.
And then once that tagged protein is broken down into its component amino acids, those are released for use by the cell. Protein degradation is important if maybe the product of a particular gene, that cell only needs a tiny little bit of it, and then it needs to mark that protein for degradation once it's done its job in the cell. A checkpoint question based on protein activation and degradation. Review figure 11.5, which is right up here. If the enzyme responsible for cleaving inactive insulin is deactivated, what effect will this have on the form and function of insulin? So if the enzyme that cleaves insulin here to form this active form of insulin is deactivated, what effect will this have? The final molecule will have a shape different from that of active insulin, and therefore will not be able to function as a hormone in the body unless it is activated correctly. So that is another way that eukaryotes can regulate gene expression. The last way that we're going to talk about is something called non-coding RNAs. Non-coding RNAs play multiple roles in controlling gene expression. One interesting fact is that only 1.5% of the human genome actually codes for proteins. This is also true of many other multicellular eukaryotes. If you look at a pie chart of all the human genome, the protein coding regions are only this little orange sliver right here. They're only 1.5%, which is a really small amount. Another small fraction of DNA consists of genes for ribosomal RNA and transfer RNA, which we know those types of RNA are needed to make the ribosomes and then for translation to occur. A flood of recent data suggests that a significant amount of the remaining genome is transcribed into functioning but non-protein coding RNAs. So it's transcribed into RNA, but those RNAs, their purpose is not to then be translated into a protein. Their purpose is just to be that strand of RNA. A variety of small RNA molecules, when bound to proteins, can prevent gene expression by pairing with an mRNA molecule and either degrading it or by blocking translation. An example of a type of small mRNA molecule that can do this is miRNA. miRNA stands for microRNA, so I'm going to draw that here. miRNA is microRNA. It's a type of small interfering RNA that can affect gene expression. So miRNA is microRNA. This is a strand of miRNA here. miRNA binds to a protein to create an miRNA protein complex. That protein complex is then what can go pair with an mRNA molecule that's complementary and either degrade it or block its translation. If we take a look at this whole process, this is what we just looked at. The microRNA needs to combine with a protein to form an miRNA protein complex. That protein complex will then go around the cell looking for any strands of mRNA that are complementary, fully complementary, or partially complementary to this microRNA sequence. The target mRNA can have a completely complementary sequence, so that means the entire length is complementary. A binds with U, C binds with G. It's fully complementary. Or the target mRNA can only be partially complementary. So in this example here, It's complementary up to this point. These 4 bases are not complementary. These 3 bases are complementary because we see the hydrogen bonds. We have 2 more bases that are not complementary, and then finally the 2 final bases are complementary, and then 1 not complementary. What the miRNA protein complex does with this specific mRNA depends on whether it's a fully complementary sequence or a partially complementary sequence. If it's a fully complementary sequence, the miRNA protein complex will degrade the mRNA, so it will fully degrade it and digest it. If it's only partially complementary, it will block translation, but it will not degrade the mRNA particle. A checkpoint question based on microRNA. If a gene has the sequence AAT, TCG, CG, what would be the sequence of an miRNA that degrades the gene? So we have the sequence AATT, CG, CG. I'm going to write that down. AATT, CG, CG. Remember, this is the sequence of a gene, so this is DNA. We need to convert this DNA into RNA. We'll make the RNA purple. So remember, A binds with U instead of T in RNA, so this would give us U, A, G, C, G, C. And it's asking us, what's the sequence of an miRNA that degrades the gene? So we know if it's going to degrade the gene, it has to be complementary along the entire sequence to degrade it. So we're going to do our microRNA in red. This microRNA strand needs to be completely complementary to this purple RNA strand. So U binds with A, U binds with A binds with U, G binds with C, and C binds with G. So this would be our complementary miRNA strand that degrades this strand of mRNA, AAUU, CG, CG. Let's see if we were correct. So the gene will be transcribed as the mRNA sequence UU, AA, GC, GC. We got that right. And then an miRNA of sequence AAUU, CG, CG would bind to and degrade this mRNA. We did get it correct. That is how you would solve a problem like that. We're going to now review the multiple mechanisms that regulate gene expression in eukaryotes. So gene expression can be regulated multiple ways within both the nucleus and the cytoplasm. And we're going to do a quick refresh of all of the ways. I'm going to go ahead and just make this figure fully and then talk through it. So we have a chromosome that is within the nucleus. The first way that gene expression can be regulated in eukaryotes is how tightly packed the DNA is. If it's super tightly packed, it will not be expressed, but if it's more unpacked, it's more likely that that gene will be expressed. The second way it can be regulated, and probably the main gene expression regulation that most eukaryotes use, is transcription, specifically the start of transcription, because all of those specific transcription factors need to bind to the enhancers to facilitate RNA polymerase binding to the promoter. If any one of those is missing, transcription cannot occur. Another way gene expression is regulated in eukaryotes is through the alternative mRNA splicing. So the exons can be combined in different ways, and that's how the 20,000 or 21,000 human genes can create over 100,000 protein polypeptides through alternative splicing. Another way gene expression can be regulated is that cap and the tail that get added on to the mRNA. Remember, certain characteristics of those cap and tails determines how long the mRNA exists in the cell. And so if it's a longer lived mRNA, it will have more protein product. If it's a shorter lived mRNA, it will have less protein product. Another regulation step is flow through the nuclear envelope. The mRNA has to be able to get through the nuclear envelope in order for it to be translated. A little sub-step here, that mRNA, how long it stays in the cytoplasm before breaking down, like we just talked about, is a way that eukaryotes can regulate gene expression. The start of translation is another way. So if translation does not start correctly, then that gene will not be expressed. Once translation has occurred and we have our protein polypeptide, that protein polypeptide often needs to be activated, whether that's through cleavage, modification, activation by bringing to a specific location in the cell. That's another way to regulate gene expression. And then finally, how long an active protein is in the cell before it breaks down is the last way that eukaryotes regulate gene expression. And when that protein breaks down, it will break down into its component amino acids. So this figure overall is all of the different ways. We have 1, 2, 3, 4, 5, 6, 7, 8, 9 different ways that eukaryotes can regulate gene expression. In prokaryotes, typically they just regulate the start of transcription for the most part. The last thing we're going to talk about in this video is how cells respond to their neighboring cells with changes in gene expression. Cell-to-cell signaling via proteins or other kinds of molecules carrying messages from signaling cells to receiving or target cells is an important mechanism in the coordination of cellular activities. So in this image here, we have a signaling cell over here. It's sending this little green circle signal to its neighboring cell, which has a receptor protein for that little green circle in its plasma membrane. Once that signaling molecule has bound to the receptor protein in the target cell's membrane, it then triggers a signal transduction pathway. We've talked about these in relation to cancer, but I want to review them again. In most cases, a signaling molecule acts by binding to the receptor protein in the plasma membrane. It does not have to enter the target cell. It just needs to bind to this protein in the membrane. After it binds, a signal transduction pathway will be initiated. A signal transduction pathway is a series of molecular changes that converts a signal on a target cell's surface to a specific response inside the cell. We're going to take a look at this specific signal transduction pathway. So the signal molecule has bound to the receptor protein. This initiates a series of relay proteins to pass on a message. This final relay protein will activate this transcription factor. This transcription factor will enter into the nucleus and transcribe a specific gene, enable the transcription of a specific gene in the DNA. That DNA will be transcribed into an mRNA sequence. That mRNA sequence will leave the nucleus and then will be translated into a new protein product. So this signaling cell has now influenced the creation of this protein product in this target cell. And remember, we talked about transcription factors just a few slides ago. Those are what help the eukaryotic DNA bend so that the enhancers can be right above the promoter and RNA polymerase can bind. That's why the transcription factors are important. And the transcription factors are pretty specific for a specific gene. So this transcription factor is specific for its gene of interest. If my explanation confused you a little bit, we have another video explanation of cell signaling.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
So one thing I want to point out is the transcription factor. It has this like half moon shape now. Before it was activated, it was just a blob. So the activation of the transcription factor changed its shape. To turn on a gene, must a signal molecule actually enter a target cell? Hopefully you automatically thought no, all it needs to do is bind to a receptor protein in the plasma membrane, and that alone will signal or trigger the signal transduction pathway. So no, a signal molecule can bind to a receptor protein in the outer membrane of the target cell and trigger a signal transduction pathway that activates transcription factors. Okay, we're going to end part 1 here, and in part 2 we will talk about the cloning of plants and animals.